Greetings and welcome back. So we're now talking in detail. We're starting to lean into the technical details associated with thinking about pattern formation, and this is a super broad topic. There's patterns in herds, there's patterns in organismal body plans, there's patterns in the proteins within a cell, where they're distributed, how a cell finds its middle, and so on. And the two paradigms that we're going to spend some time on are highlighted in this very nice paper by James Sharp. And I guess, this next couple of vignettes are going to be a bit of a James Sharp love fest. When I talk about Alistair, I always appreciate the opportunity to celebrate the contributions of Ned Wingreen and Leonid Mirny, at least to my own thinking. And here I would say that Sharp, who's at EMBL in Barcelona, has written a number of really thoughtful papers on the general topic of how spatial patterns emerge in the biological setting, usually in the context of many multicellular organism development. So this particular paper, I would consider it, in a way, required reading for this course. I think anybody that's listening to this vignette should go read it. Yes, it's a strong endorsement. Does that mean that I'm going to vouch for every single word in the paper? No, I don't have any particular criticisms to make, but I believe in the fundamental imperfection of humans, and I like to be a lover more than a hater, in the sense that I'm not that interested in spending my time criticizing other people's work so much as trying to appreciate it. And so, in this case, I thought this was a very thoughtful paper. I'm going to just read the abstract out loud. One of the most fundamental questions in biology is that of biological pattern. How do the structures and shapes of organisms arise? Undoubtedly, the two most influential ideas in this area are those of Alan Turing's reaction diffusion and Louis Wolpert's positional information. Much has been written about these two concepts, but some confusion still remains, in particular about the relation between them. Here we address this relationship, propose a scheme of three distinct ways in which these two ideas work together to shape biological form. And, I guess I wanted to comment on that, because I've found in the history of science that there are many examples of partisanship where, maybe there's two competing ideas and there's a lot of hostility as people fight for their point of view, and then in the retrospect, over the long course of history, we come to appreciate that many elements were correct from both parties. And one example of that I think is particularly cool and interesting is that of the understanding of enzyme action. So in the late 1800s, there was kind of this battle that went on between Louis Pasteur and the Buchner brothers, and you can read about this in just amazing series of articles by this guy James Barnett. So he wrote a series of articles on yeast, and if you type on his name and then you first type Pasteur and then after that type Buchner, I think you'll find the key articles. Now, why do I bring this up? Because Pasteur argued that enzymes were central to how a cell makes a living. They're lifey. They tell us about the metabolic pathways that give energy to cells so that they can do their thing, and that's good. And the Buchners said, no, this is just chemistry. You can take an enzyme and its substrate, put it in a test tube, and it'll just do chemistry. And there'll be some reaction that's catalyzed by the enzyme, and as a result, it will speed up that reaction. So, and it might, in retrospect, seem kind of bizarre that there was such emotion that was coupled to those 2 points of view. And the reason is because, at least for me, you'll have to see if you agree, but I find great pleasure, for example, in teaching freshman biology and juxtaposing and holding these two views up as complementary views of the same topic, and I don't find any contradiction between them at all. And I think it's kind of silly to get too worked up about which one's more important or whatever. I don't think that that's useful. Let me give you another example. So I just mentioned Mirny and Wingreen, and those guys inspired me to become really almost like a zealot towards the idea of allostery. There's a famous biologist, I won't mention him by name because I don't exactly know where this citation is, but the comment was made that he wouldn't even talk to physicists switching to biology unless they could define allostery. So anyway, I got super excited about allostery. It's been a decade of my life really, really thinking hard about it, and wrote a book, called The Molecular Switch, which you can take a look at. And what I came to understand was that even to this day, there's a lot of heat associated with different versions of allostery. So this is an idea that was set up in the 1960s by Monod, Wyman, and Change, and then sort of there was this parallel universe of KNF, Koshland, Nemethy, and Filmer, where they had kind of an alternative view of allostery. And like I said, there's still some heat on that, and for me, I take the perspective of statistical physics, which I'm hoping that we will bring to bear on exactly that problem later in the term. I look at those two models, and to me, they're both homework problems. They're not even different. Okay, fine, if you're a structural biologist or whatever, you might get worked up about it, but to me, fundamentally, they all correspond to the same fundamental idea, which is that a particular protein has multiple conformational states, and those conformational states have different activities, and they also have different KDs, if you like, for binding of some ligand. And so, I think my point here is that there's these two polarizing views, and what Sharp has to say is that, there's some merit to both of them, and they actually can work in unison. So let me try to explain to you what the claim is. So these are the two worldviews that we're going to spend the next week thinking about. So let's start on the left-hand side with the Wolpertian idea of positional information. So what I'm showing you on the x-axis is let's say this is the long axis, the anterior-posterior axis of some organism. The one I'll have in mind is the fly. And we're going to be interested in the development of a so-called Morphogen Gradient, where the red curve in these first four panels, these red curves correspond to the temporal evolution from back to front of the concentration of some morphogen protein such as bicoid, which is the one that we'll be interested in. And allied with that is some idea shown here that if you're to the left of this concentration, in other words, if the concentration is higher than this threshold that I've drawn here, in other words, your concentration is in this region, then you'll adopt the red fate shown on the bottom. If your concentration is between here and here, then you'll adopt the white fate. And if your concentration is lower than that, you'll adopt the blue fate. This is the so-called French flag hypothesis or model. Sharp has a very interesting article about that as well that you might find interesting to look at. But what it says is that basically there's a coordinate system that's laid down by the morphogen. What I mean by that is that molecules know, with quotes, whether they're to the left or the right of some threshold because they can measure, again I'm going to use that word, sort of anthropomorphically, they can measure the local concentration of the morphogen, and if it's higher than something, then they will adopt one fate, and if it's lower than something, they'll adopt a different fate. So this is a predisposition of molecular coordinates that's laid down in the form of the morphogen. So that's the positional information idea. Very different perspective is the self-organization idea of Turing, where the idea is that we have two morphogens around. Let's call one an activator and one an inhibitor. And as you can see, we get a little fluctuation of activator here. So there's some local increase in the concentration of activator. And what that does is, because of autoactivation, it induces more of itself. Now, that also, at the same time, the circuit responsible for this, it also starts to activate the production of inhibitor, which is shown in red. And here's the rub, the thing that's really interesting. the diffusion coefficient for red is higher than for that of green. In other words, the inhibitor diffuses more quickly. And what this means is that the red diffuses away from the scene of the crime so that it doesn't have time to suppress activator, but now it starts to bunch up in the neighboring region, and that will push down activator in that region. And so you end up getting a wave of activation and inhibition. And this is spontaneous. In other words, there doesn't need to be any pre-existing coordinate system or a pre-existing address or pre-existing concentration that specifies where the pattern will go. And we'll see later in the context of this large algal organism called Chara that the organization of actin filaments in this organism on the surface is spontaneous self-organization. One of the reasons we know that is that a beautiful experiment, it applied drugs which disturbed the actin organization, and then it would basically re-self-organize, but with certain morphological features shifted, meaning that there was no fundamental positioning of those features. So this is really just all I wanted to say by way of introduction. I wanted to give you this notion of two complementary views of the origins of patterning, and one of them is due to Wolpert and others, but that's the name that's attached to it, Louis Wolpert, and the other is attributed to Turing because of his paper, The Chemical Basis of Morphogenesis. And what we're going to do now is we're going to spend a number of vignettes playing with these two ideas. The first thing we'll do is we'll talk about the phenomenology of morphogen gradients, the French flag model and its predictions, and then I'll derive reaction diffusion equations, and we'll talk about how they're solved. So that's where we are.